Hey there and welcome. Have you ever wondered how the trillions of cells in your body just know what to do? Well, they've got this master instruction manual, DNA, but it's locked away for safekeeping. So today, we're going to dive into transcription, the amazing process cells use to make a working copy of that manual. So, how in the world do they pull that off?
So that really is the big question, right? If the master blueprint, your DNA, is way too valuable to ever leave the super safe cell nucleus, how does the cell actually build all the stuff it needs, like muscles or enzymes, way out in the main part of the cell? Well, it needs a messenger, right? A working copy. And that is exactly what transcription is all about. Okay, so this diagram here basically lays out the cell's entire game plan. Biologists have a fancy name for it, the central dogma. It might look a little complicated, but the main idea is actually super simple. The master plan, DNA, is used to create a temporary message, which we call RNA. That step is transcription. Then, that RNA message is read to build a protein, and that's called translation. But for today, we're laser-focused on that critical first step, getting the message out of the DNA and into an RNA molecule.
Okay, so to really get transcription, you gotta know the two main players here. The best way to think about it is this. DNA is like the original, priceless master blueprint locked away in an architect's vault. You'd never take that to a messy construction site. RNA, on the other hand, is the photocopy you make. It's a temporary working copy that's designed to get the job done and can handle a little wear and tear.
And this table breaks down the differences perfectly for us. You can see DNA has that famous double helix structure, while RNA is just a single, more flexible strand. Chemically, they use almost the same language, but with one crucial difference. DNA uses a base called thymine, or T, while RNA swaps that out for something called uracil, or U. And here's the kicker. For its job, DNA always stays safe inside the nucleus, while RNA is built to travel out into the rest of the cell.
All right, for your exam, if you remember nothing else, remember this. DNA is double-stranded and uses thymine, T. RNA is single-stranded and uses uracil, U. That T for U swap is a huge deal. It's like the signature of an RNA molecule, a dead giveaway that you're not looking at DNA.
Okay, let's get into the nitty-gritty of the process itself. How is that RNA copy actually made? It all happens in three pretty straightforward phases, and if we stick with our analogy, you can think of it just like using a photocopier. There's a beginning, a middle, and an end. Simple!
These phases are called initiation, elongation, and termination. Initiation, that's just loading your document and smacking that big green start button. Elongation is the machine actually scanning the page and printing out the new copy. And termination is when that fresh copy slides right out of the tray, ready for you to grab. See? Easy as that.
So let's zoom in on step 1, initiation. Now don't get distracted by all the stuff happening in this diagram. We're focused right here, on transcription. The cell's photocopier is a really important enzyme called RNA polymerase. It's this little machine that scans along the DNA until it finds a special Start Here sign called a promoter. Once it finds it, it locks on, unwinds a little bit of the DNA, and gets everything ready to copy.
Next up, we've got elongation. And this is really the main event. The RNA polymerase enzyme just zips along one of the DNA strands, using it as a perfect template. As it chugs along, it reads each DNA base one by one and grabs the matching RNA piece to build the new messenger RNA chain. It's this incredibly fast and super precise assembly line, building out the message letter by letter.
And that brings us to the final step, termination. The polymerase just keeps on building that RNA strand until it hits a specific DNA sequence that basically screams, "Stop, we're done here." This stop signal makes the enzyme let go of both the DNA blueprint and the brand new, freshly made messenger RNA copy. The job is done, and the message is ready to go.
But wait, how does the polymerase know which RNA letter to add? You see, making an accurate copy is everything. The cell can't afford any typos. To get it right every single time, the enzyme follows a very strict set of pairing rules. And honestly, getting these rules down is your key to acing this topic on your test.
All right, here are the rules of the road. Cytosine, or C, will always pair up with guanine, or G. That one's easy, C with G. The twist comes with A and T. When the DNA template has a T, the new RNA strand gets an A. But, and this is the big one, the one everyone gets tripped up on, when the DNA has an A, the RNA strand gets a U, for uracil. Why? Because remember, there's no T in RNA.
Okay, listen up, because this is the most common mistake I see students make on tests. Your brain is going to scream at you to pair an A on the DNA with a T in the new strand. It just feels right, but you have to fight that instinct. Just burn this into your memory. RNA does not have thymine, so an A on the DNA template always pairs with a U in the RNA. I guarantee you'll see this on an exam.
So, need a simple way to make this stick in your brain for the test? Try this little trick. For the A to U pairing, just think apples under the tree. And for the C to G pair, think cars in the garage. They might sound a little goofy, but trust me, these memory tricks really work when you're under pressure.
All right, so we did it. We have our messenger RNA copy. But now what? Where does it go? Let's zoom out for a second and see where transcription fits into the cell's entire protein-building factory.
Here's the whole shebang, from start to finish. Step 1. The DNA blueprint stays protected in the nucleus. Step 2. Transcription. The process we just covered happens right there to make that mRNA copy. Step 3. That new copy leaves the nucleus and travels out into the cytoplasm. Step 4. It finds a little protein factory called a ribosome, and that's where translation happens. And finally, step 5. A protein is built. Transcription is that absolutely essential first domino that has to fall.
So, just to bring this all home with our main idea, on the left you see the biological reality of what's happening, and on the right you see a simple office photocopier. One is complex biology, the other is a machine, but the principle is exactly the same, making a portable, working copy from a protected master document. And at the end of the day, this is why transcription is so incredibly important. Your genetic code, your DNA, is just information. It can't actually do anything on its own. Transcription is the process that activates that code, turning that silent, static information into a living, breathing action plan for your cells.
Which, of course, leaves us with one last huge question. We now have our messenger RNA copy, this long string of A's, U's, C's, and G's. But how does the cell read that simple four-letter code and translate it into the incredibly complex, three-dimensional machinery of a protein? But that is a story